Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about Ara Operon or Arabinose Operon. In the previous video's lecture we talked about Rna Operon, and before that we talked about the lactose operon or lac operon. We also talked that what is lac operon, how it is activated, how it is deactivated, how it is switched on and switched off. We also talked that how Rna operon is switched on and switched off. We talked these two operon in detail. Moreover, we also talked that lac operon is actually positive transcriptional. It is a positive transcriptional regulation process, while Rna operon is the negative transcriptional process. Okay. Negative transcription regulation process. Then in this video we are going to talk about Arabinose Operon or Ara Operon, which is actually positive as well as negative transcriptional control. Now how we'll be talking in this video, so please stay tuned and keep watching. Okay friends, so now we are back. Now what is Ara Operon? So let me write. Okay. Now what is Arabinose Operon or Ara Operon? It is actually a set of genes which contain Ara structural genes and Ara regulatory genes as well as Ara promoter and each and everything. Okay, which mainly produce the Arabinose enzyme, and after that these Arabinose enzymes are used for different purposes, or it can usually take the Arabinose, converted the Arabinose into Xylose 5-phosphate, and then it is used for the cell wall synthesis. So this is actually Arabinose Operon. What is actually Arabinose? First of all, let's talk about that. This Arabinose is actually bipolymer. Okay, this is the bipolymer, let me write, that can make the cell wall. Okay, now how it can make the cell wall? This Arabinose is also Aldopentose. Now why we call it Aldopentose? Because it contains 5 carbon atom, and moreover it is also act as a aldehyde functional group. It have aldehyde functional group, that's why we call that aldehyde functional group. Okay, so if we see the structure of this Arabinose, it will be look like in this form. So this is actually what, this is the Arabinose, and usually this is L-Arabinose. Okay, it is actually how it can make the cell wall. This is a question arise. This Arabinose is then converted into D-Xylose 5-phosphate. How it will be look like? It will be look like in this form. Okay. So now this is actually the structure of Xylose 5-phosphate. Let me write. Phosphate. In short form we can write like that. And this is L-Arabinose. Okay. So when we get the Xylose 5-phosphate, then this is actually the main component of the cell wall. When we have Xylose 5-phosphate, then the cell wall will be formed. This cell wall, this molecule is mainly involved in the plant cell wall. Okay, this Arabinose is what? This Arabinose, remember guys, this is again a disaccharide, or it is a carbohydrate again. Now, so this is a little bit about the intro of the Arabinose. Now then we have Arabinose Operon. Remember guys, in the previous video, if you guys remember, we talked that lac operon was the positive regulation of transcription. It gave positive signal for the transcription regulation. And we talked that Rna operon is the negative, it gave negative signal for the regulation of the transcription. This Arabinose is what this is this Arabinose mostly give positive and negative signal. This give positive and negative signal to the regulation of transcriptional control. Now how? Because if we it give positive and negative signal due to the presence of Arabinose or absence of Arabinose. If we have Arabinose or if we don't have the Arabinose, then it gives positive and negative signal to the regulation of the transcription respectively. This is actually how we can say that mostly it contain why we say that it is positive or negative, because it contain, we have in this case we have a protein which we called it Ara C protein. Ara C protein. This Ara C protein is actually involved to switched on and switched off. This Ara C protein is actually perform two function. It either give positive signal or negative signal in the presence of Arabinose or in the absence of Arabinose respectively. That's why we can say that it is both positive and negative transcription, regulation of transcription. Now let's talk about that Arabinose operon function, that how it is switched on and switched off. In the previous video we talked that trap operon is always switched on. Naturally it is switched on, and like operon is always switched off. Okay, but in this case it is sometimes it is switched on and sometimes it is switched off. Switched on, switched off. Normally it's switched on and switched off. Switched on and switched off. But how? Let's talk about that. First of all, we have to talk the main components which are involved in the Arabinose operon. Then we will explain that how it is switched on and switched off. We have main components. So the first main components that are involved in this case we call that Ara C protein. Now how Ara C protein will look like? Ara C protein have actually three main structure. They contain three main parts. We have first part, let me draw. This is the first part and this is the second part. Remember guys, this first part is actually DNA binding site. This second, as you can see, this is actually linker. This is what this is, linker. And this third, as you can see, this is the Arabinose binding site. So this is the structure of what Ara C protein. Then we have another main component, we called it. So second we have Rna Polymerase. So we already talked about the RNA structure, but here I am drawing a simple structure of the Rna Polymerase, as you can see. So this is the structure of the Rna Polymerase. Although this is not the real structure, but somehow Rna Polymerase will look like in this one. Then we have Rna Polymerase. Third we have cap and camp complex. Okay, complex. Again, how this protein will look like, we also talked that we have this. Let's suppose this is what, this is the cap protein, and upon it we have another protein, we call that camp. Okay, so this is what, this is actually cap complex. We have this too. So these are actually the three main proteins that are involved in the Arabinose operon. Now how the Arabinose operon will look like? Let's draw the structure of Arabinose operon. So okay friends, this is overall the structure of Arabinose operon. Let me a little bit explain. This Arabinose operon contains Ara O, means Arabinose operator genes. It contains Ara C, means Arabinose C protein genes. It contains promoter for Arabinose C. So remember, most of the time we talk that in lag operon and in trap operon, the promoter is present at this, means at upstream. Okay, while this operon is operator region is present downstream. But in this case, promoter is present at downstream of this operator and this Ara C protein. So Rna Polymerase will sit here and will move at that direction. It will move upstream. Remember guys. And in this case, this is actually the process. In this case, the process will be occur in this way. And then we have Ara O, means Arabinose operator 1. We have cap binding site, means cap and cap complex will bind at that site. We have Ara I, means Arabinose inhibitor site. Then we have promoter for BAD genes, BAD genes are actually, these are the genes that are mainly responsible to convert the arabinose, to take the arabinose and make it into xylose 5-phosphate to make the cell wall. Okay, so these are the three genes that perform that type of the function. Mainly these genes produce polycistronic mRNA, and this polycistronic mRNA perform that function. After that we have these genes, and at the end we have terminated sequences where the RNA polymerase will detach from there and the process is completed. So remember guys, the DNA are actually in a linear form. Okay, DNA are not in a bent form as I draw. This is not in a bent form. There is no space. That's why I draw some portion of the Arabinose operon at that under this. Okay, now what kind of enzymes are produced from these genes, and what kind of function these enzymes perform? So remember guys, this B produce enzyme, we call that ribolokinase. This A produce Ara isomerase. The third gene produce ribulase 5-phosphate epimerase. Ribulose 5-phosphate epimerase. Okay, so these are the most important enzymes that are produced by these genes. First of all, these genes are converted into polycistronic mRNA. After that we get translation occur. After translation we get these three types of the protein. These three types of the protein mainly perform different function. Respectively, they perform different function. These protein actually perform the metabolism of xylose 5-phosphate. Okay, they perform the metabolism of xylose 5-phosphate and make the cell wall. At that time when we have arabinose. So this is the this is actually the operon. Now this is all about the Ara Operon. Now how it is switched on and switched off. This operon will be switched on at that time when there is glucose. Let's suppose if we have glucose present, if we have glucose absent and arabinose present, at that time this process, this operon will be switched on. Now how? Remember guys, this will be switched on by the absence of glucose and presence of arabinose, because this glucose and arabinose give signal to this promoter C. Now once the promoter C is activated, so then it will bring what it will bring, RNA polymerase. RNA polymerase will sit here. So when RNA polymerase will sit here, it will transcribe the Ara C genes. It will be transcribed in upstream. Okay, it transcribes the Ara C genes. So then we get what? We get the Ara C genes, as you can see. This is the Ara C mRNA, or we can say polycistronic mRNA. Not polycistronic, Ara C mRNA, let's suppose. From this we get the Ara C protein. Okay, so what we get after that? We get the Ara C protein. Now after that what happened? We have also the cap and cap complex. If you guys remember, in the lag operon we talked that if we don't have the glucose, we have the camp concentration is high. Okay, the camp concentration is high. Again in this case we don't have glucose, so camp concentration is high, which it will bind to the cap. So what we have the camp concentration get high. If it get high, so it will bind to this and we get cap complex, as you can see. This cap complex will bind to this cap site. So let's suppose at that point we have what we have, cap complex, which are bind. After that what happened? Remember guys, this DNA is in a linear form, not in a bending form. Okay, now after that what happened? We have Ara C protein that are activated. Then arabinose molecule will bind to this active site. Remember guys, this is Arabinose molecule. It binds to this active site or to this site of this Ara C protein. So once it is bind, so then what we get? We get two Ara C protein in a dimer way, in a dimer form. And in this, with this two Ara C protein, the Arabinose molecule are binded. So when it is binded, so these two dimer are bind. One dimer are bind to this Ara O site, while the other dimer are bind to this Ara I site. Okay. So let's suppose we have what? We have the, let's suppose this is what? This is again the, so as you can see, we have Ara C protein having what? Having the Arabinose. It will bind to this site. And some Ara C protein will bind to this. So remember guys, if this Arabinose if is present and Ara C is also present and making a complex, as you can see, at that way what happened? Rna polymerase is attracted and Rna polymerase will sit here. Rna polymerase is attracted and sit here, as you can see, and transcribing the genes. So it will transcribe the genes and produce what? Produce the polycistronic mRNA. And this polycistronic mRNA give rise these three types of the protein, as you can see. So this is actually in a linear form, and this RNA polymerase are bound at this way, and it will transcribe the gene in downstream. Remember guys, okay, at this promoter C region, it will transcribe the genes at upstream. This is very important. There is no space, that's why I draw it under. Okay, don't be confused. So this is how the Arabinose operon get activated. Then how this process, Arabinose process, will be deactivated. So it is activated by this Ara C protein. Again it is deactivated by this Ara C protein. Now how? Remember guys, if we have glucose, how it is deactivated? If we have glucose but we do not have Arabinose, at that point this process get deactivated. Now how this glucose presence and Arabinose absence cause the switched off of this Ara Operon? Remember guys, if we have glucose and Arabinose is not present, so we have the camp concentration get decrease. Okay, camp concentration get decrease. So if we do not have camp, so we do not have this cap protein, or we do not have camp cap complex. If we do not have camp cap complex, it will not bind to this area. If it is not bind to this area, what happen? RNA polymerase will be not attracted. Moreover, what happen? We have Ara C protein. This Ara C protein, if there is no Arabinose, remember guys, so this active site is free. If this active site is free, so then what happen? Remember guys, if this active site is free, this Arabinose dimer will make a bond with each other. How? Let's suppose this is one Ara C and this is another Ara C. As there is no Arabinose between them, so then it will make interaction with each other, as you can see. So this is the dimer. So this dimer will bind, then what happen? The DNA will be bent. The DNA bend, and once the DNA bend, so then RNA polymerase again not attracted, because the way for the RNA polymerase will be stopped. And this gene is transcribing, but the RNA polymerase is not, RNA polymerase is just binding at that point. And if it is binding at that point, so this gene is activated. It is not binding at that point. Why? Because these two dimers are binded here, and it causes the bending of the DNA, due to which the space are not available for the RNA polymerase. Now how? So now how this Arabinose will be deactivated? So remember guys, first of all what happened? First of all, we have these two Ara protein. This two Ara protein, actually it is not in a bending way. First of all, it is in a linear way. After that, once it is in a linear way, so then what happened? We have the Arabinose, this Ara C protein. This Ara C protein is actually one Ara C protein bind at this point, while the other Ara C protein bind at this point. Okay, so when these two Ara protein, in which one bind to this Ara O while the other bind this Ara I, once it is bind, so then what happen? It will cause the bending of the DNA. As you can see, DNA will bend. So when the DNA is bent, so what happen? These two active site of this Ara C protein will interact with each other, as you can see. That's why the bending is formed. Moreover, sometime what happen at this Ara O, there is also this 2 protein. Okay, we have also the Ara C protein at this Ara O site. Okay, so remember guys, if the Ara C protein are bound to at that specific point of the DNA and the DNA will be bent, so then what happen? After that, RNA polymerase will be not binded. Although the CAMP complex is not present, that's why the RNA polymerase will be not attracted, because these two complex means CAP complex, actually it will attract the RNA polymerase. And if it is not bind, so then what happen? Transcription will be not occur. If transcription is not occur, we this messenger RNA is not formed, which we do not get these 3 enzymes, and at that way there is no formation of this Xylose 5-phosphate, and cell wall is not formed. This is all. That's how it is deactivated. Now we have what? We have this RNA polymerase. It is not bind. Moreover, this CAMP complex is also not bind to this CAP site. Okay, there is also a CAP site which is available at that point. So if these things are not binded, so RNA polymerase is not bind, and the process will be stopped. Ara Operon will be switched off. So this is how the Ara Operon is switched on and switched off by this one protein, Ara C protein. Okay, friend. Now there is a question arise in our mind. Let's suppose if we have glucose and we have Arabinose, then what happen? There is a question arise. If we have glucose and Arabinose, so then what happen? The CAMP complex, this CAMP molecule get decreases, and we do not have the CAMP complex formation. At that point, what happened? The Ara C protein are formed because Arabinose is present. So Arabinose will bind to this Ara C site, and it will bind to this Ara O and this Ara I site. Still the transcription is occur. Why? Because the space is free. Although the CAP complex is not present, but still the transcription is occur. But remember, at that point the transcription is a very occur, but it is very occur at a low level. Okay, transcribe the genes at a low level. Second, remember guys, if we have no glucose and we have no Arabinose, then what happen? This CAMP complex, this CAMP molecule get increased, and it will make the CAP complex. This CAP complex will bind to this CAP binding site. It will bind to this binding site. After that, these 2 protein, Ara C protein, again bind, one protein bind to this Ara O while the other bind to this Ara I, and bend the DNA. If it bend the DNA, so then other 2 dimer of Ara C will bind to this Ara O site. So if the bending occur, again the RNA polymerase not bind. Although we have what? We have CAP complex. Let's suppose this is what? This is the CAP complex, but still the transcription will not occur because it is halted by these 2 protein. Okay, so this is how the presence and the absence of the glucose and the presence of both glucose or Arabinose, or the absence of both glucose or Arabinose, will affect on the switching on and switching off of the operon. Moreover, the most important protein is Ara C that are also involved in switching on and switching off of the Ara Operon. Okay, I hope you guys get idea about that. If you like this video, so make sure to hit the like button, share this video, and also subscribe the channel and press the bell icon, okay, to get notification. Thank you so much for watching that video.